Hi, Dr. Wang here. Today we're going to use the CMAP software to make a flowchart for the FOA assay experiment. The lab instruction is available on the Commons page. Please go to Book Lab, Week 11, FOA assay, and we're going to do this virtual experiment in Week 12. In this experiment, we start with 3 yeast strains. So these are the AGY75 yeast strain transformed either with the wild type PMSH2 plasmid, the mutant PMSH2 plasmid, or PRS413. So those are USP0, SP1, and SP2, 3, 4, or 5. The first step is to use the OD1 yeast overnight culture to prepare 2 additional dilutions. So the first dilution should be at OD 0.1 and the second dilution should be at OD 0.01. The necessary calculation for this is in assignment lab 9.1, and you're supposed to write the calculation in the flowchart as well. So here is the starting material, the 3 yeast strain overnight culture at OD 1.0. And from this starting material, you're going to make the first dilution. So this is at OD 0.1. So here, this is a C1, V1, C2, V2 question, right? So here, this is the first concentration, C1, and then the question mark, how many milliliter of this to use? This is V1, and then over here, C2 is 0.1, and V2 is 1 milliliter. You're going to make 1 milliliter of a diluted yeast culture at OD 0.1. So optical density is equivalent to the concentration of the cell in the suspension. So once you solve this question mark using C1, V1, C2, V2, you're also going to figure out the amount of water to use. So this 2 volume combined should give you 1 milliliter total. So here, these 2 numbers together should add up to be 1 milliliter. So here's how to solve this question. And then you're supposed to fill in all the question marks in this table that are up here. So once you prepare the 9 dilutions, you're going to plate the 9 yeast dilutions on the plate. This is a template for how to plate the 9 yeast dilutions on the plate. So you're going to plate them, the 3 yeast strains in rows, right? So here, yeast transformed with the wild type, the mutant, or the empty vector plasmid. And at the 3 different yeast dilutions in columns. So here, this is OD1, OD 0.1, and OD 0.01. Coming back to the CMAP, so once you have the 9 yeast dilutions, you're going to centrifuge, discard the supernatant, and then add 100 microliters sterile water to the yeast pellet. So here you have 100 microliter yeast suspension. Half of those will be plated on SD plate using the template, and the other half will be plated on FOA plate using the same template. The growth pattern on FOA plate will tell you the mutation rate in the 3 yeast strains. And the reason that we're plating this yeast at 3 different dilutions, 1, 0.1, and 0.01, is so that we have a range to capture the optimum density that will allow us to tell the difference in growth pattern between the 3 yeast strains. What you measure in the end in this experiment is the dependent variable. Okay, so what does it represent? To think about the independent variable, let's come back to this template. So here, the independent variable is basically the difference between rows, right? So for example, what is the difference between the samples that are in the first row and the samples that are in the second row, or the samples that are in the third row? Also, what is the difference between the samples that are in different columns, right? So if we fix this variable right here, just look at the samples within the first row. What is the difference between this cell and this cell? So that's the second independent variable. There are two independent variables in this experimental setup.